Mic check.
Mic check.
Hello, guys. Oh, welcome to Antibuddies. This is 21st Journal Club in our series, and we have three guests with us actually. First, we have got Cole Delia, who is a YouTuber in the Investigate, Explore, and Discover YouTube channel. And then we've got our guest who is going to present us a beautiful paper about T cell signaling, Dr. Deborah Yablonski. And let me introduce her a little bit before we start with the discussions. Uh, Debbie is a preclinical faculty member at the Rappaport Faculty of Medicine of the Technion Israel Institute of Technology, where she heads the Immune Cell Signaling Lab and teaches immunology. At the Immune Cell Signaling Lab, she seeks to decipher intracellular regulatory mechanisms that precisely control immune responses, specifically in T cells and mast cells. As part of her work, they have identified regulatory junctions that depend on GRB2 family adapter protein known as GADs. The lab is currently working to determine the regulatory functions of GADs in an in vivo mouse model and to apply these insights into developing new immunomodulatory approaches. In her free time, Debbie and her family enjoy music, hiking, and dance. So, right before we start, Debbie, can you tell us what kind of music do you like?
Oh, I like so many kinds of music, and my daughter also loves to sing, so we love to hear her singing. Um, yeah,
Okay.
not a
Right.
very
Yeah,
specific answer,
yeah. A
but
lot of times we don't hear the other side of the scientists. Like they have personal lives, right? They like music, they like dancing, just like every one of us. So it's good to hear about those things once in a while. But yeah, Debbie, thanks a lot for joining us today for our journal club. And with that, I will transfer the mic to you. Please feel free to present.
thank you. Thanks. So, like I said, I'll try to pause for questions, but if I forget to, just jump in with
Mm-hmm.
questions. I don't mind. So today I'm going to present a study. We have the first author of the study, Inas Halumi, here with us today as well, which is very nice. And we published the study recently in the Journal of Immunology. Um, so I want to start just by actually, this is not working. I want to start by introducing my lab members. These are the current members of my lab. Inas, who's with us today, Rose, Shalach, who's the second author on the paper. I want to introduce my collaborators, Arthur Weiss and Wanlin Lowe from UCSF, and Henning Urlaub and Jasmine Corso from the Max Planck Institute in Germany. So I want to just thank everybody before I start so I don't forget. Very important. And anybody who's interested to hear more about our research, you're welcome to visit our website, which is listed here. So basically, the question that kind of drives all of our research is we want to understand how T cells integrate information from their surroundings and just basically how do they decide what to do. Okay. And for people who may be a little bit less familiar with T cell receptor signaling, I just thought I'd start with a very basic introduction. And of course, everything starts with the T cell receptor. The T cell receptor is the clonotypic receptor. You see it here. It kind of resembles an antibody. I know you guys are the antibody club. And it kind of resembles the FAB region of an antibody in that the T cell receptor has a variable region that's specific for each clone of T cells and a constant region. But you'll notice that the T cell receptor is always membrane bound. And the reason for that is that T cells always recognize antigens in the context of a cell-cell interaction. That's why they're the cellular part of the adaptive immune system. And you can see here that the antigen recognized by the T cell receptor is always a peptide that's presented by MHC on another cell. So that's what T cells recognize. And virtually at every stage of their development, the activity and the development and the differentiation of T cells are governed by the signaling through their T cell receptor. Okay. And we know that the signal that's experienced by the receptor can basically be strong or weak, depending on how high the affinity is. And we know that T cells experience thresholds. So those of you who've studied immunology know that within the thymus, above a certain threshold of TCR activity, cells are considered to be autoreactive and are removed from the repertoire by negative selection. And below that threshold, the cells are considered to be safe and potentially useful for our immune response, and they develop, they undergo positive selection and exit to the periphery. Okay, this means kind of weak recognition of self-antigens. In the periphery, the situation is reversed. This weak recognition of self-antigens induces tolerance or homeostasis, whereas stronger recognition may induce an immune response. So the idea of being able to recognize strength of signaling is very important, but we know that it's not the only factor that governs T cell responses, because in addition to sensing the level of signaling, here you see the kind of cell-cell interaction of a T cell with an antigen-presenting cell. And you can see that in addition to the level of stimulation through the TCR, T cells are also affected by the presence of a co-stimulatory signal received through CD28. And this is sometimes determined the danger signal. If the cell is giving a co-stimulatory signal, it means that it's seen some kind of microbe or experienced some kind of dangerous situation, and it's telling the T cell, you better respond to this message that I'm sending you. But of course, the T cell doesn't always respond. In order to respond, it has to receive both signals. It has to both see the danger signal received through the co-stimulatory signal received through CD28, and the antigen-specific signal received through the T cell receptor. And this creates a kind of additional decision-making point where the T cell will decide whether to remain tolerant or, if both signals are present, to trigger an immune response. Now, as we'll see, one question that's not that clear is how do the T cells integrate the information from those two receptors as they decide whether to respond? So let's start with the T cell receptor signaling pathway. And I brought one slide that's just from an immunology textbook, just to kind of remind everybody what we're talking about. And then we'll move on into our more specific research. So again, everything starts with the T cell receptor. But the T cell receptor itself, you'll notice it has this rather large extracellular region that recognizes the antigen, but you see these chains, almost nothing in the cell. So how does it transmit the signal into the cell when it binds antigen? Well, it does this through these accessory chains, which are called CD3, and there's a number of accessory chains that you can see here. And they all have this motif containing two tyrosines called an ITAM motif. And why do I stop to talk about this ITAM motif? It's not that important for my talk today, but it's important for immunologists because the same motif occurs on quite a large number of immune receptors, whether it's B cell receptor, FC epsilon receptor, other excitatory receptors, they all signal through these ITAM motifs. So what happens when the T cell receptor is triggered? We get by peptide MHC, see here the peptide MHC on another cell, we get recruitment, we get a cascade of tyrosine kinases, which begins with recruitment of the Sark family tyrosine kinase, LCK, that phosphorylates the ITAM motifs. And now we have a signal inside the cell that activation has occurred on the outside, okay? Another thing we have inside the cell is a second tyrosine kinase, the second tyrosine kinase in the cascade known as ZAP70. Now, ZAP70 is a pretty poor tyrosine kinase, and it's normally found in an inactive conformation. It's very low basal activity, but it does have these two SH2 domains that are perfectly situated to bind to a doubly phosphorylated ITAM motif. So once these ITAMs are phosphorylated, ZAP70 can bind, switch into its active conformation, and to begin to phosphorylate downstream substrates. So we have now a cascade of 2 kinases, right? LCK and then ZAP70. And in a way, we can say that the strength of signaling through the TCR has kind of been transduced into the strength of ZAP70 activity. Okay? Now, what does ZAP70 do? ZAP70 phosphorylates 2 of the stars of our show today. 2 adapter proteins. One is membrane bound and is called LAT. And the other is cytoplasmic and is called SLP76. It's easier to say SLP76, and that's how I'll refer to it. So from this whole chain of events, we end up with phosphorylation of 2 adapter proteins. I can't remember which of you said that you work on adapter proteins or protein-protein interactions, but the interesting thing about adapter proteins is that they have no enzymatic activity, and yet they're able to exert very strong effects on signaling pathways through their ability to perform protein-protein interactions, okay? And once these adapter proteins get phosphorylated on tyrosines, phosphotyrosine becomes like an anchoring site that can bind to specific proteins through their SH2 domains. So we're going to get the building of a large complex. And in the next slide, I'd like to show you how that complex gets built around LAT and SLP76. So here we see, I have a little animation here. So as I said in the previous slide, the TCR ligation gets transduced into the activation of ZAP70, and ZAP70 phosphorylates 2 adapter proteins, LAT, we see here membrane bound, and SLP76, which is cytoplasmic. Both get phosphorylated on particular tyrosines. And then we have a 3rd adapter protein, which is called GADS, okay? For those of you familiar with GRAB2 adapter, GADS is from the GRAB2 family, and it's characterized by an SH2 domain surrounded by 2 SH3 domains. Sometimes I used to like to call it the Mickey Mouse family of proteins, because when you draw it, it looks like Mickey Mouse with the SH2 and the 2 SH3s on either side. So this is a different representation of the same thing we saw in the previous slide, but we're going to take it one step further. Because what happens when LAT and SLP76 gets phosphorylated? GADS, through its SH2 domain, is going to bind to LAT. And thereby, it recruits SLP76 into the LAT complex. Now we have a complex, not separate proteins, okay? And other phosphotyrosines here, we have a lot of them, recruit other proteins. So we get a huge complex. I'm only going to show you part of it. It's actually much bigger than I'm showing you. And for that reason, it's sometimes called a signalosome, kind of like ribosome or whatever. This is a signalosome. And it gets formed when T cells are stimulated. So here we see the enzyme phospholipase C, gamma, gets recruited through one of its SH2 domains. Other signaling proteins get recruited to SLP76 through their SH2 domains. I'm just showing them very simply here, but these are also big complex proteins. VAV and NCK, which have activities that we don't have time to talk about today, but we're going to focus on the 3rd protein here, ITK, okay? ITK is also a tyrosine kinase. So notice we have a cascade here of 3 tyrosine kinases. We had LCK that phosphorylated the T cell receptor, recruiting ZAP70 that phosphorylated LAT and SLP76. Can you see my mouse when I point with the mouse? Good. And they recruit a 3rd tyrosine kinase, ITK, which is from the TEC family. In previous work in my lab years ago, we showed that the activity of ITK actually depends on its binding to SLP76. So the active ITK can be found in this complex, Okay. Now, what does it do within this complex? What ITK does is it becomes active, as I said, and it phosphorylates PLC gamma at sites that are required for its activity. So we have this huge complex, and right now it looks like a machine for activating PLC gamma, you know, which used to confuse me when I presented this work, because I would ask myself, why can't ZAP70 just activate PLC gamma? Wouldn't it be so much easier? You know, why do we need to make this huge machine just to activate PLC gamma? So I think, you know, we now know that the answer to that is that it's a very important thing that happens within this complex, but it's not the only thing. This signalosome is coordinating a lot of different aspects of the T cell response. But let's just look for now at phospholipase C gamma. If you're familiar with phospholipase C, you know that it hydrolyzes a membrane phospholipid to create 2-second messengers. And these are called inositol triphosphate and diacylglycerol. So now we have 2-second messengers. Inositol triphosphate induces calcium flux, which brings about the activation of a transcription factor known as NFAT. I call it NFAT. Okay. And diacylglycerol can bring about activation of RAS pathway, which activates another transcription factor called AP1. Okay, so this is all very complicated, I know, a lot of words, a lot of names, but just consider that I've taken you actually from the membrane to the activation of transcription factors that are involved in the transcriptional response to TCR stimulation. And one of the most important aspects of the transcriptional response is transcription of IL-2, of the cytokine IL-2, because that's the growth factor for T cells, right? What happens when a T cell first gets activated? It starts proliferating like crazy, right? Are you guys familiar with that? That's what one of the reasons why your lymph nodes swell up when you get sick is because the B cells and T cells that were activated are proliferating like crazy. And one of the main factors that helps T cells to enter this proliferative state is that they begin to transcribe and translate and secrete IL-2, which acts as a growth factor. So we've really kind of traveled down the whole pathway. But one of the things that interested us when we looked at this years ago, as I said, it's kind of a big complex just to activate PLC gamma. And so we thought there must be other regulatory events going on within the complex. So one of the ways we decided to look at this is we isolated from TCR stimulated cells, we isolated GADs and SLP76, and we asked, are there any other phosphorylation events on these proteins that maybe we haven't considered yet? So we did a phosphoproteomic analysis, and we found 2 phosphorylation sites, both of which turned out, you'll see in our talk, to be mediated by ITK. So ITK is not only phosphorylating PLC gamma within this complex, it's also phosphorylating SLP76 at tyrosine 173, and it's phosphorylating GADs at tyrosine 45. Okay, now here, this is a log 2 base scale. So you see that the increase in phosphorylation upon stimulation is really huge. It's not a 5-fold increase, it's a 2 to the 5-fold increase, which is quite a large number. So ITK very strongly induces the phosphorylation of these proteins. And we wanted to know, why do we need these additional phosphorylation sites? What do they do? Okay, so I want to kind of give you the bottom line at the beginning of the talk, and maybe that will help you to follow the talk. What we found is that these 2 sites help to integrate the T cell receptor pathway that I already showed you about with the second pathway I told you about, which is CD28 co-stimulation. Okay, and for this, it's very important to think about IL-2 that I told you about before, because the unique thing about IL-2 is that even though I just described the whole T cell receptor pathway leading to its transcription, TCR stimulation is not sufficient to get transcription of IL-2, it also depends on CD28 co-stimulation, which brings about the activation of NF-kappa B that binds to this CD28 REP element that has to be activated in order to get IL-2. So IL-2 is kind of the distal point at which TCR and CD28 co-stimulatory activity are integrated. But we really didn't know how the integration occurs further upstream, how the integration of this information occurs further upstream. And this is one of the subjects of our study. Are we okay till now, Jen?
Yes. I have one question. It's a very basic one. So I know the SH2 domains bind to phosphotyrosenes. What do SH3 domains do?
Okay, good question. So SH2 and SH3, they were both first discovered in the tyrosine kinase SARC, and that's why they have that name, SARC homology 2 and SARC homology 3. And the SARC homology 1 domain is the kinase domain. The SARC homology 2 domains, there's about 100 of them in the human genome, and they all bind to phosphotyrosine, but each one has its own phosphotyrosine motif that it likes to bind, depending on the surrounding sequence. So those are inducible interactions. SH3 domains tend to have constitutive interactions. So they bind to particular peptide motifs, but it tends not to be inducible. So for example, here the C-terminal SH3 of GADS binds very strongly and very specifically to SLP76. Okay, now often these SH3-mediated interactions are proline directed. So there's certain motifs, PXXP, that people like to talk about. But some of them, for example, the interaction between GADS and SLP76 is not proline directed. It binds to an RXXK motif. So in my mind, I tend to give long answers to short questions. But if
That's completely fine.
If I want to give a short answer, SH3 domains bind to proline-rich motifs. The longer answer is in those SH3 domains that have really high affinity interactions, the motifs tend not to be proline-rich motifs, but tend to be other motifs. So it's kind of an open question. But the interactions are generally constitutive. The SH3 interactions are generally constitutive.
I had one more thing, just for a recap. So LAT, GADS, and SLP76, none of these are kinases. They're just adapters sticking to each other when there is a phosphorylation on LAT.
Yes, it's an inducible complex of adapter proteins. And there's even more adapter proteins in the complex. But yeah.
Okay.
Any other questions before we go on? Now, I have to apologize because I wasn't able to kind of unify my presentation of these proteins. So you're going to see different presentations. But a lot of our work focuses on GADs. So I want to show you just another representation of this complex that kind of gives us a better idea of the complex in GADs. So this is the same complex. I apologize, it's a different representation. This is one that Inas made, and it's so beautiful. I had to include it. And what you can see here is this, first of all, the similarity between GADS and GRAB2. Both have the structure of a central SH2 domain surrounded by two SH3 domains. GRAB2 also binds to LAT, but its role is to recruit SAS, a RAS exchange factor, to LAT. So GRAB2 recruits SAS to LAT. GADS looks very similar, but it recruits SLP76 to LAT. Okay, so that's one thing you can see. You can see that the C-terminal SH3 of GADS binds tightly to this motif I mentioned a minute ago, RXXK motif in SLP76. It's very tight binding. And another thing that we showed in a study I don't have time to talk about today, this was shown by another member of my lab, is that GADS actually binds cooperatively to LAT. Actually, GADS dimerizes, and a pair of GADS molecules binds to these two sites on LAT, tyrosine 171, phosphotyrosine 171, and phosphotyrosine 191. So for a second, let's just see what that might look like. You actually have everything here on the right side occurring in double. I lost my mouse. I can't find my mouse, but can you guys see what I'm, yeah.
Yeah.
Everything here occurring in double, because you have two GADS molecules binding cooperatively to LAT. And this has the effect. Now, remember, I'm drawing this, Inas drew this in 2 dimensions, but it's a 3-dimensional complex. So you can imagine that as part of this 3-dimensional complex, ITK, which is recruited by SLP76, gets brought very close to its substrate, phospholipase C gamma, which is also bound to LAT. And in this way, it helps ITK to phosphorylate phospholipase C gamma at 783. But another thing you can see here is this N-terminal SH3 domain of GADS, which appears to be doing nothing, right? Doesn't bind to anything. It doesn't have any function, and there's actually no known function for this domain, except now we know it's phosphorylated on tyrosine 45 upon TCR stimulation. So let's take a look for a minute at this evolutionarily conserved. Hey, Cole, you want to ask a question?
Yeah. So similar to Jatin's question about the other protein interacting, when ITK gets, or does ITK dimerize for one if it's in double or in duplicate in this system?
Yeah, we haven't looked at that directly, but it may, and a lot of kinases, their activation is assisted by dimerization. So I don't think that would be a problem, but we haven't really looked into that in any kind of way.
Okay, no worries. And then if you haven't looked into the interaction of ITK dimerization, would you assume, since most of these proteins function is assisted by dimerization, would you think that that influences how it interacts with PLC gamma?
Yeah, I think it's possible. And the other thing you have to keep in mind, and I didn't bring this up here, is that I'm just showing you like one LAT molecule, but actually LAT is known to form microclusters when the TCR is stimulated. And those can be mediated by SAS, actually, because each SAS molecule binds to 2 GRAB2 molecules. And so those can bring in other LAT molecules, and each SLP76 there's another adapter that binds to 3 SLP76 molecules, and LAT binds to 2 GADS molecules. So you can see how this kind of becomes like a web, you know?
Mm-hmm.
And you can see this web, you know, we just look, it's easier for us to just look at one kind of unit of it, but it's actually a web that forms a microcluster.
Okay. Awesome. Thank you for that.
Okay. Should I go on? Yeah. Okay. So I want to focus for a minute on this N-terminal SH3 domain, which is very conserved. As you can see here, we have just kind of like alignment of different species, from human to trout, xenopus, all different kinds of vertebrate species. And you can see that for a domain that has no known function, it's very conserved. So that kind of high level of conservation doesn't really sit well with having no function. And also the tyrosine phosphorylation site itself, and this I forget what you call these kind of diagrams, but this diagram is based on more, I think like 85 species or something, and shows the conservation around the phosphorylated tyrosine 45.
Are they called web logos?
Web logos, yeah.
It's a very weird name. I have no idea why they came up with that, but yeah, something like that.
In any case, the size of the letter indicates the degree of conservation, and you can see that it's quite high. And this is, if I'm not mistaken, among more than 80 species. So we have a highly conserved phosphorylation site in a domain with no known function and no known binding partner. And the fact that there's no known binding partner is not for lack of trying. Okay, so to study this, we made use of a genetic system in which we made a GADS deficient Jurkat-derived T cell line. And Inas and other people in the lab reconstituted these cells with different forms of GADs, all strep tagged so that we can follow them, and either wild type lacking the Y45 site, lacking the N-terminal SH3, and you'll see others also in the course of the presentation. And we also, with the help of a company, made a phospho-specific antibody against phospho-Y45. So using these reagents, we could begin to understand how the site is behaving in cells. And so first of all, in these cells, which were reconstituted with wild type GADs or GADs lacking the Y45 site, you can see that our reagent can identify TCR-induced phosphorylation of the site, and we can also identify the site just with a regular phosphotyrosine antibody. And this signal is mostly diminished when we mutate the site, meaning that this is the main tyrosine phosphorylation site on GADs. So that's in human. In mouse, we had help from Wanlin Lo from Art Weiss's lab, who showed that in naive CD4 T cells or thymocytes, we can see TCR induced, or this is actually TCR CD28 induced phosphorylation of GADs at tyrosine 45. So, and yet another representation of the complex. Here you see the dimerized GADs bound to SLP and ITK, and GADs is getting phosphorylated to tyrosine 45. Now we, based on the sequence and some other clues, we kind of guessed that ITK may be the kinase phosphorylating it. So to test this guess, Inas purified active ITK from TCR stimulated cells. She used GADS deficient cells, so she wouldn't have any GADs, endogenous GADs in the assay. And she mixed the active ITK with ATP and recombinant GADs, just to see if ITK can phosphorylate GADs. And the answer is yes, it can. We got phosphorylation of wild type GADs on tyrosine 45, but using this reagent, no phosphorylation of mutants of forms of GADs that lack this site. Okay. Now again, we could see phosphorylation of other sites in this in vitro kinase assay, but we don't see them in cells. So we think that tyrosine 45 is the main tyrosine phosphorylation site of GADs, and it's the one that we focused on for our research. So this slide shows that GADs Y45 can be phosphorylated by ITK in an in vitro kinase reaction, but is ITK the kinase that phosphorylates GADs in cells? So for that question, we used an ITK inhibitor, which abrogated phosphorylation of GADs Y45, SLP76, 173, and phospholipase C gamma, known ITK phosphorylation site. So all these three sites are phosphorylated by ITK. We also used an ITK deficient Jurkat cell line that was generated by Wanlin Lo in the Weiss lab. And again, all phosphorylation of all three ITK targets was diminished, both the GADs targeted site, the SLP76 targeted site, and the phospholipase C gamma targeted site. So based on these data, we know that GADs Y45, SLP76, 173, and PLC gamma are all phosphorylated by ITK upon TCR stimulation of intact T cells. And we kind of wanted to know, okay, if ITK, SLP76 and GADs are bound to each other, and ITK was phosphorylating both of them. So do these events have anything to do with each other? So to start to address these questions, we used a different cell line, a jerk-derived cell line lacking SLP76, and again, reconstituted with wild-type SLP76 or SLP76 lacking the ITK targeted site. We okay till now?
Mm-hmm.
Okay. So do these sites, form a linear pathway, or are they independent of each other? So here's some kind of initial data. It's a little bit confusing, but bear with me. We have here TCR-induced phosphorylation events in these blots, right? All the blots have a common structure. We have the phospho blot on top and the total protein blot on the bottom, okay? So here we see in wild type cells, inducible phosphorylation of GADs, Y45, and we see the same thing in cells that lack the SLP76 phosphorylation site. So you don't need phosphorylation of SLP76, 173 to get phosphorylation of GADs, okay? And vice versa. If we take cells that are either wild type or lacking the GADs tyrosine phosphorylation site, in both cases, we still get tyrosine phosphorylation of SLP76. So ITK can phosphorylate both of these adapters independently of each other, separately from each other. And not only that, I have like a pile of negative data, Inas and I and other members of the lab, we have a pile of negative data that we're not going to show you. We're going to spare you. But just suffice it to say that it was very difficult to find out what these phosphorylation events are doing, because our assumption was we consider the signalosome to be a machine to activate PLC gamma, and we couldn't find any effects on PLC gamma or events directly downstream of PLC gamma, as hard as we may look. And here's a sample of this kind of data. If you mutate Y45 of GADs, you get perfectly fine phosphorylation of PLC gamma. So we kind of, showed that ITK phosphorylates these sites, but we were a little bit mystified, and the sites are evolutionarily conserved. They're phosphorylated in human and mouse. Why? We were kind of mystified why. So it's not part of the pathway leading to phospholipase C gamma. So before we get to what it does do, I want to just show you how ITK phosphorylates GADs. How does it happen? Because we know, I showed you that ITK, binds to SLP76, and it has to be bound to SLP76 to get activated. And then it kind of makes sense. Active ITK is always bound to SLP76, so it phosphorylates SLP76. It's right next to it, right? But what does it need in order to phosphorylate GADs? So here in this representation, you see the dimeric GADs bound to LAT, right? And the rest of it is shown only once. So you have GADs bound to SLP76, SLP76 bound to ITK. Here's phospholipase C gamma. It's the same molecules, a little bit different representation. And first of all, we wanted to know, do you need this whole complex for ITK to find GADs and phosphorylate it? So we kind of disrupted the complex at different points. First, Inas made a cell line in which the interaction between GADs and SLP76 was disrupted by a mutation in the C-terminal SH3 of GADs. So we just made a mutation. The GADs is there, the SLP76 is there, but they can't bind to each other. And now what's going to happen? So what happened is that we completely lost the interaction of GADs with SLP76 as expected. We didn't lose phosphorylation of SLP76, but we completely lost phosphorylation of GADs. You see that here? Mm-hmm. We completely lost phosphorylation of GADs. So meaning that ITK sits on SLP76 and phosphorylates it, that's fine. But in order to get to GADs, it needs GADs and SLP76 to be bound to each other. Okay, it really needs GADs to be brought to it by binding to SLP76. So what about lat? Do we need lat? So here, again, we used cells that Wan Lin generated, which lack lat. So now we have GAD, SLP, ITK, all those things are there. Again, SLP76 is still phosphorylated. So SLP76 is kind of easy. It just needs to have TCR stimulation. It binds to ITK, gets phosphorylated. But GADS is more difficult. It needs to be bound to lat, and it needs to be bound to SLP76. It needs the whole complex. Okay? So from these experiments, we conclude that GADS Y45 phosphorylation occurs within the SLP-GADS lat complex.
I have a question here.
Yeah.
Yes, this result is when lat is not there, right? So without lat, how does ITK get the signal from the TCR?
Because SLP76 gets phosphorylated even in the absence of lat. We don't have it in this thing. The three N-terminal sites of SLP76, one of which recruits ITK, those can be phosphorylated in the absence of lat because ZAP70 can phosphorylate SLP76. And then phospho-SLP76 can recruit ITK, and ITK can get active.
Okay.
And bear in mind, we're probably doing very strong stimuli here. So maybe that's also part of the reason. But even with these strong stimuli, if GADS can't bind to lat, or if GADS can't bind to SLP76, it doesn't get phosphorylated.
Okay, thank you.
So it kind of seems like a flag almost, when GADS gets phosphorylated, it's like a flag went up and it says, you know what, there's a signal going through here. It's real, right? Because the whole complex was created, and it's kind of like a type of signal that TCR signaling has occurred. Okay, that we kind of tried to look at it that way. And so that generated kind of a working hypothesis that the ITK targeted sites, their purpose is to help to integrate TCR and CD28 co-stimulation, to kind of set up a signal that says, you know what, TCR stimulation has occurred, everything's okay on our side, so if there's co-stimulation, let's go ahead with this. Something like that. And I haven't really shown you the evidence for that, but I'm kind of, but this is kind of what our evidence suggests. So I want to show you some of the evidence for that, and we're okay with time. Okay. So for this part, we used what are called reporter assays, in which we have various transcriptional elements driving the expression of a luciferase reporter gene. So we use two different reporter assays. One is the NFAT reporter that can be activated just by TCR stimulation alone. And the other is the CD28 REAP reporter that requires both TCR and CD28 co-stimulation to be activated. So here's the results. When we look at the NFAT reporter, you can see it's activated. Let's look here. In the absence of SLP76, it's not activated. But when you have SLP76, it can be activated by the TCR or by TCR CD28 co-stimulation. Okay? And if you don't have the 173F site, it's fine. In some experiments, it's a little bit lower. In some experiments, it's the same. In general, it's fine. So you don't need 173F for force signaling through the TCR pathway, PLC gamma, to the activation of the NFAT transcription factor. And you also don't need the 45F site for this, okay? I told you we have a pile of negative data. This is part of it, part of the pile. Now let's get to the positive data when we look at the REAP motif. The graphs look different, right? Let's look at SLP76 first. Again, no SLP76, no activation of this motif. Wild type SLP76, you get activity upon TCR CD28 co-stimulation. And if you don't have that 173F site, nothing. Okay, so you need this site to somehow respond to the co-stimulatory signal or to integrate in the co-stimulatory information. Same thing for the 45F site. It seems to, the result is less dramatic, but again, it seems to contribute to the co-stimulatory responsiveness. So the results were kind of different, right? Because here you see SLP76 is required for REAP responsiveness, whereas GADS itself wasn't actually required for REAP responsive. GADS is much more confusing than SLP76. So at this point, we decided to take it step by step. Let's figure out what the SLP76 173F site is doing in this pathway, and then maybe that'll give us a clue to understand what the GADS site is doing, okay? So are there questions till now? No. So we're going to just start with the SLP76 site and the GAD site, and then we'll be all done. So again, this REAP motif is part of the IL-2 promoter. So we looked at actual IL-2 production in these cells, and the hatched bars are in the presence of TCR CD28 co-stimulation. And this is the percent of cells that became IL-2 positive. And you see that the percent is significantly decreased when cells lack the 173 tyrosine 173 phosphorylation site. The results are much cleaner when Inas did a qPCR, quantitative RT-PCR reaction. And so here you see upon co-stimulation, the induction of IL-2 mRNA in wild type cells, and it's virtually eliminated if you just mutate this one tyrosine phosphorylation site. So it really seems to play a role in the ability to respond to co-stimulation. Okay. Now, NF-kappa, this REAP site binds to AP1 and NF-kappa B. We focused on NF-kappa B, and indeed using an NF-kappa B reporter plasmid, we saw, this is a luciferase reporter plasmid, the 173F mutation. It gives us a reduction in activity of NF-kappa B. So it seems that SLP76 tyrosine 173 is selectively required for an NF-kappa B dependent branch of the TCR signaling pathway, which responds to both TCR and CD28 co-stimulation. So how does this NF-kappa B family pathway work? We know that NF-kappa B family members like CREL are held in the cytoplasm by an inhibitory protein, I-kappa B. And upon stimulation, I-kappa B gets phosphorylated, ubiquitinated, and degraded, and releasing the NF-kappa B family member, which can now translocate to the nucleus and activate transcription. So this is, I think, subsequent to the publication of our paper. Inas looked at these factors. She looked at I-kappa B phosphorylation and degradation, and published in the paper, she looked at CREL translocation to the nucleus. And she got beautiful results. Here you see phospho I-kappa B induced by TCR CD28 co-stimulation is reduced by the 173F mutation. Here you see quantification of this data. Degradation, here you see degradation of I-kappa B after 60 minutes of TCR CD28 co-stimulation in the wild type cells, but not in the 173F cells. And as a result, since the I-kappa B is phosphorylated less and degraded less, you get, less CREL accumulating in the nucleus. This is the band to look at. This is TCR and CD28 co-stimulation. Here you have TCR, CD28 co-stimulation of wild type cells. You get CREL accumulating in the nucleus. And here we have TCR CD28 co-stimulation of the mutant cells, much less CREL accumulating in the nucleus. The band to the right of these is a positive control in which we use PMA and ionomycin to bypass the requirement for SLP76, and then all 3 cell lines accumulate CREL in the nucleus. So all 3 cell lines can translocate CREL, but in order to do it in response to TCR and CD28 co-stimulation, they require tyrosine 173. Here you see quantification of these results. So again, it's a significant effect. But you know, the really surprising result is we also had less CREL accumulating in the cytoplasm. Because when you look, Inas, is this hours here? It's hours, right?
Yes.
Yeah. So when we looked 4 hours, here, these are all minutes, but when we looked 4 hours after TCR stimulant, oh no, these are hours, maybe 2. When we looked 4 hours after stimulation, the wild type cells have a higher level of CREL in the cytoplasm, and the 173F cells don't. And this, I think, reflects the ability of CREL to trigger its own transcription, kind of turns itself on. So to look into that more, Inas did a qPCR looking at the transcription, the amount of CREL transcripts in the cells. And you see, Inas, what time point is this? 4 hours?
Yes, also 4 hours.
4 hours. So 4 hours after TCR CD28 co-stimulation, we have a huge increase in the amount of CREL transcripts. And this is reduced in the 173F cells. So this is kind of a real lot of data. And even almost for myself, I just kind of made a list of what we've seen here. So what we see is that the 173 tyrosine phosphorylation site, even though it's on SLP76 and it's considered a canonical part of the TCR signaling pathway, it affects the ability to respond to CD28 co-stimulation. And it does this by affecting the NF-kappa B pathway at multiple levels. It's required for I-kappa B phosphorylation, I-kappa B degradation, leading to nuclear translocation of CREL, all of which leads to transcriptional induction of CREL, which in turn is required for transcriptional induction of IL-2. Okay, so these results kind of explain on a mechanistic level the selective requirement for SLP76 tyrosine 173 to integrate TCR and CD28 co-stimulation. And to the best of my knowledge, it's I'm afraid to say the most, but it's a very proximal point of integration of TCR and CD28 co-stimulation. I'm not aware of a more proximal point of integration.
Debbie, there's one thing for, I don't know if you already mentioned this and I just missed it, but what's the link between SLP76 and IKV alpha? Is it going through the IKK? Or is there another kinase that's...
I don't know.
Okay.
You didn't miss it. We don't know.
Okay.
I would love to know.
There's also another question from the YouTube audience.
Yes.
Okay, do you hear me? Okay, so the question is, do you think that PI3K signaling from CD28 is important for activity of ETK, phosphatoid trikinase? is it important
Is
for the
PI
activity?
is PI3 kinase required to activate
Yeah.
ITK? I
Yeah.
think it is, because
Mhm.
ITK has a pH domain, but I have to say we worked very hard. We wanted to try to show that phosphorylation of these sites, SLP76, 173, and GADS-Y45 actually depends on CD28 co-stimulation, as you're suggesting, that CD28 co-stimulation activates PI3 kinase, and that would be required to activate ITK and phosphorylate these sites, and we could not show it, not in Jurkat cells and not in primary mouse cells.
Oh.
In both systems, the sites can be phosphorylated by TCR stimulation alone. And that's, I didn't show that here, but that's our findings. And that's why what we think is going on is it's not that CD28 is required to phosphorylate these sites. It's actually that phosphorylation of the sites is required to respond to CD28
Oh.
in some way. It's the opposite. Okay?
Mm-hmm.
It's a good question. Any other good questions or bad questions? Any kind of question? Okay. There are no bad questions, of course. Okay, so now let's go to GADS-45. Here we have, it's more difficult to explain what's going on. And the part of the difficulty is that GADS itself seems to be less important in these assays. So here, for example, we see the nuclear translocation of CREL upon co-stimulation. So these are wild type cells. I apologize for the jargon. These are cells that lack SLP76, and we don't get nuclear translocation of CREL. And these are cells that lack GADS, and they're fine, you know? Also, the cytoplasmic induction of CREL is fine in cells that lack GADS. Also, as we showed before, the activation of REAP, transcription reporter, is fine in cells that lack GADS. So you don't really need GADS to do any of these things, but if you lack one little phosphorylation site on GADS, it doesn't work. Okay, so this is like majorly confusing, and the model that we came up with to explain this is that GADS is a bifunctional protein. It has positive functions and negative functions. Okay, so the positive functions are easy to understand, right? Because through its SH2 domain, it binds to LAT. And through its SH3 domain, it binds to SLP76. So it brings SLP76 to LAT, and it helps makes this whole complex, the signalosome that transmits TCR signaling. You know, that's easy. The negative signaling function, we think, relates to this mysterious N-terminal SH3 domain that doesn't seem to do anything. And now we think that what it's actually doing is restraining responsiveness. And it's kind of like, we can almost think of it as like a gate. So in unstimulated cells, the gate is closed. If we're talking about the gate to reach REAP activation. Okay, in unstimulated cells, the gate is closed. Unstimulated cells don't activate REAP in any of the cell lines. Okay, when cells are wild type and they're stimulated, the gate opens and REAP can be activated. Part of the opening of the gate appears to be removal of the inhibitory function of the N-terminal SH3 of GADS, which gets phosphorylated, and this appears to remove an inhibitory function. Okay, if we don't have any GADS at all, there's no inhibition, and so we can walk through the gate. There's no door, there's no blockage, and so once the TCR stimulation comes along, we can walk through it. But if we have mutated GADS that has an increased inhibitory function, then even if we stimulate the cells, we can't reach the activated state. So this is kind of a complicated model. I hope I explained it well. the gist of the model is that GADS not only has a positive signaling function, it also has a negative signaling function. And the negative signaling function we think is carried out by the N-terminal SH3, and we think it needs to be phosphorylated to remove this inhibition. When GADS is not there, we lose both functions. It's like kind of like having a car that loses the gas and the brakes. So you can't really control it very well, but if you push it, it'll go. That's kind of how we look at cells that lack GADS. So we know, as I told,
Sorry,
yeah?
could I just make sure that I've got that final part correct with tyrosine 45? Like, could you say that it effectively acts as a key so that like the tyrosine 45 site needs to be phosphorylated for GADS activity to function, but without that site, without that like keyhole, effectively the door remains shut?
Yeah, something like that.
It
Okay.
is a very complicated model. Like, I can imagine when you were coming up with, we had these results, it would have been so hard to imagine what kind of system is this. It's really not a typical on and off switch system.
It took us a long time. It was very frustrating, and I really appreciate that my students didn't give up, you know? So one support for this model, what we thought is it's very confusing to work on a protein that has both positive and negative signaling functions. But we have GADS mutations that can't do the positive signaling functions, because as part of another study, we created mutant forms of GADS that can't dimerize. And if they can't dimerize, they can't bind to LAT. And if they can't bind to LAT, they can't do the positive signaling function. So maybe in those mutants, we'd be left with only the negative signaling function. So we took a look at those mutant forms of GADS. Here we have two of them, two different mutations that impact the dimerization domain. So they cannot bind to LAT. Here you see in a co-immunoprecipitation, when you precipitate GADS, even though the cells have phospholat, the phospholat is not associated with GADS because they have a dimerization domain mutation. They can't bind to the phospholat, okay? And the GADS does not get phosphorylated. So these were very interesting mutants because, I'm sorry for jumping back and forth, they lost the positive functions of GADS. They can't dimerize, they can't bind to LAT, they can't get phosphorylated at tyrosine 45, and maybe they have only the negative functions left. So what happens with these mutants when we put them into the REAP luciferase assay? They have very low activity, okay? And we saw even lower activity when we did a, again, you don't need GADS for this assay. These are cells that lack GADS. These are cells that have wild type GADS. You don't need GADS for this assay. But if you have non-dimerizing, non-phosphorylated GADS, it interferes with the response. We saw this interference even more strongly in a transient assay. Here we have increasing amounts of transfected GADS. So wild type GADS has a positive role, and it can increase activity in this assay above that seen in the absence of GADS, whereas the non-dimerizing mutants of GADS had a negative role in this assay. As we transfected it in, it reduced the response to below that seen in the absence of GADS. So here you see the opposing positive and negative regulatory functions of GADS. When we just mutate the tyrosine 45 in GADS, we get kind of an intermediate response. So to summarize, we've shown you that there's TCR-induced ITK-mediated phosphorylation of GADs, Y45, which occurs only when GADs is found within the lat nucleated complex, because it has to bind both to SLP76 and to lat to get phosphorylated. And we saw that also SLP76 tyrosine 173 can be phosphorylated by ITK, and it's required for activation of CREL, translocation of CREL to the nucleus, transcription of CREL, and for REAP transcriptional activity. And we saw that non-phosphorylated monomeric GADs selectively inhibits this REAP pathway. So those were our main conclusions. This is the graphical abstract that accompanied our paper, and it's numbered according to these conclusions. There's a lot here that remains uncertain. For example, as I can't remember who asked, was it you, Jatin? How do SLP76 and GADs even regulate CREL? It seems to be independent of their effects on phospholipase C gamma, and how does this occur? Another important question is, how important is GADs 45 phosphorylation in vivo? Because we saw it's a very conserved site. It hasn't to date been studied. And so we generated a mouse model lacking GADs tyrosine 45, and we're just really starting to analyze that mouse model and try to see what's the function of the site in vivo. We know that ITK itself can influence CD4 T cell subset differentiation. That's one of the in vivo roles of ITK. So we really want to know what part of these effects are mediated by its ability to phosphorylate SLP76 and GADs, and when we don't have anything close to an answer yet. So that's kind of the end of my talk. I, of course, want to again thank my lab members. Here's another picture, an older picture of lab members. In addition to Inas and Rose, who are in the previous picture, you also see Dvora Beach, who was first identified the tyrosine 45 site through a mass spec study. You see Sam Whitman, who did a lot of the luciferase assays. And Amy helped us with some of the assays. She's a medical, they're medical students, helped us with some of the assays. You can see our collaborators from UCSF, Arthur Weiss and Juan Lowe, in our funding. And of course, I'm happy to answer any more questions. And I'm really kind of glad to have this opportunity. It's so unique to have this kind of student-directed seminar. I think it's very special.
Thanks a lot, Debbie. Thanks a lot for being here today. I think I have learned so much about T-cell signaling in this session that I'm not going to forget it for a while. Because in the textbooks, we only have this surface interactions, just things that are barely required. But in this one, it's so detailed. And at the same time, I feel good that I thought I don't know a lot about T-cell signaling, but generally, we don't know exactly how these elements interact with each other. And it's still a work in progress.
Mm-hmm.
I had a question from one of the, somebody asked about PI3K signaling and CD28. So I was branching off from that question. I would think that since ITK has this plex-strain homology domain that allows it to dock to certain phospholipids, and PI3K creates those phospholipids. So I would think that ITK does not function unless it's docked. Is that true? Or does it have a function even when it's not docked to the membrane?
So much of our work was done in Jurkat cells. Mm-hmm. And Jurkat cells lack PTEN. That's part of how they're transformed. The PTEN is the lipid phosphatase that opposes the activity of PI3 kinase. So since Jurkat cells lack PTEN, they may have a higher basal level of PI3 kinase products than normal T cells. And in fact, that's what if that's how they're immortalized. If you transfect them with PTEN, they die. So I'm always a little bit reluctant to answer any questions that relate to PI3 kinase based on my experience. Having said that, in primary T cells, which have PTEN and they're not transformed, we also found that Y45 of GADs and Y173 of SLP76 were phosphorylated by the T cell receptor alone and did not require CD28 co-stimulation. So that would suggest that you don't absolutely need CD28 mediated activation of PI3 kinase to get ITK activated. I think it's possible, since SLP76 recruits ITK basically to LAT, basically it recruits ITK to the membrane, right, or very close to the membrane by binding to LAT. Right? I mean, SLP76 binds to GADs, GADs binds to LAT, LAT's in the membrane, ITK is bound to SLP, all this kind of brings ITK to the membrane. Maybe that's enough to release its pH domain. I don't know. There's also reports of PI3 kinase being recruited to LAT. So maybe there's a little bit of PI3 kinase activity in LAT, and that does the trick. Basically, a very long way of saying, I'm not really sure. It's a good question.
I have a question about GAD's function. You, in your data, you mentioned that other tyrosines on GADs were being phosphorylated. Did you investigate any of those other sites further to see if those might be important for its function?
So we have two indications that other sites in GADs might be phosphorylated. One is a phosphomass spec analysis of GADs that was purified from stimulated T cells, and that revealed tyrosine 45 and another site whose number I can't remember. And the other indication is when we took ITK and allowed it to directly phosphorylate GADs in vitro. And that revealed tyrosine 45 and I think 3 other sites. Enos probably remembers this better than I do, but none of them were the same as the one that was phosphorylated in cells. So since we had this jumble of other sites that were inconsistent with each other, the only thing consistent was that tyrosine 45 was phosphorylated in both settings, and tyrosine 45 was the only one of them that was very highly conserved. And so we focused on that.
Do you think any of those other sites could possibly have function, or is it just unlikely because they weren't seen together? In your
we didn't decide to pursue them. We didn't pursue them, so I can't definitively say. But also, if you look at, there's something called the phospho. I forget what it's called. There's a website where you can kind of see every phosphorylation site that's ever been identified. Phosphosite, I think it's called Phosphosite or Phosphosite 2 or Phosphosite Plus, something like that. It's a wonderful website, and you can see every phosphomass spec analysis basically that's been done and what phosphorylation sites were identified. And tyrosine 45 of GADs was identified in dozens, or maybe well over dozens of studies, but it was never functionally characterized. And the other sites that we saw, you also can't see them on phosphosite. So we think we're pretty safe in saying that tyrosine 45 is the main one.
Okay, awesome.
What's the next step for you guys in this line of work?
So we made a 45F mouse, which was a big investment for us. And looking at that mouse, we want to look for, we want to compare it phenotypically to ITK deficient mice. Since ITK is the kinase that phosphorylates, and just try to figure out what maybe subset of ITK phenotypes might be mediated through GATs phosphorylation. That's a big part. And another part of it is looking more closely at the transcriptional events that occur upon TCR stimulation. So doing kind of quantitative PCR comparisons of different transcriptional events.
So if the whole mouse has this mutation, would you think there might be something different with the thymic selection? Maybe more Tregs or less Tregs because you're changing the threshold.
Still looking at that. I can't really, it's all these mouse studies are very early days.
Okay.
And I can't really say with any kind of confidence what phenotypes we're going to see, but I'm very excited to do these studies.
Yeah. I would say anything that changes the strength of TCR signaling should have an effect on that, theoretically. So yeah, that would be interesting. But yeah, Magda, Yang, do you guys have anything?
I didn't hear you.
Right. I really like this. Like I like the idea that some proteins doesn't need much, and some proteins have be phosphorylated in a particular place, in a particular site to be activated, and other doesn't need much, and still they work. So it's very interesting that they're so different. Yeah. That's my thoughts, but no questions from me.
Okay, then I think this would be a good time to wrap up this session. Thanks a lot, Debbie and Enas, for joining us today. And thanks to everybody who is watching. And this video will stay on YouTube and Facebook for later watching. We'll see you guys in 2 weeks for our next career talk. We have Pedro from Pedromics, the comics. He's joining us to talk about his experience as a comic creator and all sorts of things. So yeah, we'll see you again in 2 weeks. Everybody, thanks a lot for joining us.
Thank you. Bye.